Hello students, good morning. Welcome to another biology session from Anand Medical Academy. Let me continue the topic of human health and disease. This time you are going to learn about human immunodeficiency viral infection, that is AIDS. What is exactly the full form of AIDS? Acquired immunodeficiency syndrome. So today in this class what I am going to exactly talk about the AIDS. It is worldwide. The first case identified in the 1981. Over 25 million of people are dying over the year ages. Till date, so many people in India and out of India also are being infected by the AIDS. It is also sexual transmitted disease. Some important points and available things about the AIDS I am going to teach you. So, AIDS is caused by retrovirus. Viruses are many groups: myxovirus, paramyxovirus, retrovirus, flu viruses, so many. COVID-19 is also a retrovirus. In that, genome is RNA. Here also, genome of the virus is RNA. Guys, as of you know very well, viral particles consist of either DNA or RNA. Apart from that, they have a protein code. So, according to the genome, viruses are two types: DNA viruses and RNA viruses. As RNA is the genome of HIV, it is RNA virus, and it is also group of retrovirus. Now, shape of this virus is icosahedral shape, and structure, if you observe the structure of the HIV, just see, it has two single stranded RNA molecules, and reverse transcriptase enzyme is also attached or associated with the genome, and this structure is covered by a protein coat, and entire this structure is covered by another protein coat, and all this complex structure is covered by a lipid coat. So this is lipid coat and this is protein coat, secondary protein coat. Inside that, primary protein coat is there. So too much of protection, very tough. Then what are these two? This one and this one. These are single stranded RNA fragments, molecules, and along with that, the attached things. This is reverse transcriptase enzyme. Generally, in the molecular base of inheritance, we have seen that transcription from the DNA, mRNA has come. For that, transcriptase, RNA transcriptase enzymes are required. But here, RNA becomes DNA. So this is reverse transcription. For that, enzyme is required, is called reverse transcriptase. We can say this is the weapon of the HIV. With help of this enzyme only, RNA becomes DNA in the host body, means in man. Apart from that, isomerase, first enzyme, second one is reverse transcriptase, third one is polymerase. These three enzymes are also present in HIV virus. Then the first protein coat of P is 17, nothing but molecular weight of that protein coat is 17, whereas secondary protein coat is called P24. So P17 is the primary coat, which is around the two single stranded RNA, whereas single stranded RNA plus P17 together covered by another coat, that is called secondary protein coat, which is called P24. Now third coat is absolutely lipid coat. Lipid coat consists of glycoprotein membranes. are glycoprotein receptors. Glycoprotein receptors are like this. So what you are observing is a complete structure of HIV. So this is how glycoproteins this is how glycoprotein receptors are present. They are called GP120. Between GP120, other glycoprotein receptors are present. They are shorter than that. They are called GP41. Like this. This is GP means glycoprotein. So here P17 means protein code 17, whereas this is protein code 24. But last surface is not protein code, lipid code. On the lipid code, glycoprotein membranes are present. So this glycoprotein G120 and this is glycoprotein 41. Two types of receptors are present. Entire this structure from here to here, entire this structure is called capsid. So protein coat or outer coat other than the genome, virus minus genome is called capsid. The capsid of HIV virus is too tough. It consists of two protein coats and one lipid coat. On the lipid coat there was two types of receptors are present. Mainly these GP120 receptors are very dangerous. They target our T helper cells mainly. Then the T helper cells function will become 0. As T helper cells role is present in both humoral immunity, B lymphocytes providing defense, cell mediated immunity, T lymphocytes providing immunity, the T helper cells role is very important. But undoubtedly the GP120 receptors are killing the T helper cells. Then B lymphocytes immunity, humoral immunity, T lymphocytes immunity, cell mediated immunity, cell mediated immunity, both will collapse. Then ultimately immune system deficiency starts. So most dangerous thing is GP120 in the AIDS virus structure. So this is a basic data. Let me continue the topic with infection and incubation. When this HIV virus infected to the man, how does they infect? They infected through sexual transmit, nothing but sexual contacts, nothing but the person who is victim of AIDS participated in the sex, then this virus will be transmitted. And also by injections, the persons who are addicted to the drugs, they share the injections from person to person in friend circle. So one person who is the victim of HIV, the same needle will be injected without change, then HIV transmits. Also through placenta from mother to child. These are the ways where infection generally takes place. So this is HIV virus. The structure already you have seen. You imagine that HIV. Once that HIV comes, it has a GP120 receptors. Main receptors are GP120, and inside they have a single stranded RNA as well as 3 enzymes. What are the enzymes present in the HIV virus? Isomerase, reverse transcriptase and polymerase. Main enzyme is reverse transcriptase. So when they come, first they attack on monocytes, and they also attack on T lymphocytes. But in monocytes, what is going to happen? Let us see. If this is the monocyte is nothing but a type of WBC which do not have a granules, so that it is called agranulocyte. Agranulocytic leukocyte means WBC. As this virus is coming inside, it has a large nucleus which is mango shape, and the genome of our body is DNA, but the genome of virus is RNA. Once this RNA virus, nothing but retrovirus, HIV, infects monocytes, they are also called macrophages. Generally they provide immunity in our body by phagocytosis. So these are RNA virus enters inside that, they release the genome, entire virus do not enter, only the genome will be released. This is what? RNA. This RNA cannot incorporate with the DNA. Why? Because in RNA, adenine, guanine, uracil and cytosine is present, thymine was absent. Whereas in the DNA, AGTC, adenine, guanine, thymine, cytosine is present. As the base sequences of RNA with DNA cannot be compatible, these cannot, what happens? Cannot incorporate. But RNA is transcribed into DNA in host cells by the help of enzyme is called reverse transcriptase. Reverse transcriptase is also present in this virus that associated with the RNA. That RNA is injected into the macrophages, nothing but monocytes. Inside the monocytes, after infection, the RNA of the HIV virus converts into DNA in host cells by the help of reverse transcriptase enzyme. So DNA to RNA transcription, but RNA to DNA reverse transcription. Reverse transcriptase enzyme do not provided by our cells. It brought along with the genome of RNA. Once DNA is formed, this DNA is incorporated, means adjusted with our monocytes DNA. Means now this is not monocyte. Monocyte is turns into virus. It is also called provirus. This provirus undergo multiplication and produce so many cells. All these cells having DNA, but all this DNA is not have a genetical makeup of human, genetical makeup of RNA. That's why all these are called proviruses. Proviruses are coming from monocytes. These all proviruses again affect to the next fresh monocytes, and the monocytes are becoming proviruses as this viral load increases. Our monocytes, primary defensive system monocytes number will be decreases. Then our body become viral factory. So this is a factory. It will become a factory of HIV. So for this, a person required 5 months to 10 years of time. Not so simply, not so next day or next week or next month. So during this stage, a person cannot show the symptoms. That's why it is called asymptomatic period. It is also called incubation period. What is incubation? Incubation is a period or a duration between infection and symptom. Until unless a person shows some symptoms, entire the infection stage is developing, pathogenicity occur, that is called incubation. What is the incubation period of HIV? 5 months to 10 years. Sometimes maybe 1 year or 2 year or 3 year, according to our immune capacity, it exceeds. But once a person is infected by HIV, not very next month he can be a positive. In this stage, this person is not showing any symptom. It is an asymptomatic period. On the other hand, T lymphocytes were there. T lymphocytes having MHC2 receptors, means major histocompatibility. Major histocompatibility. These are the type of receptors. They help us to recognize foreign bodies. But this GP120 receptor directly attacks the MHC2 receptors of the T helper cells. They are also called CD4 cells or T4 cells. CD means cluster differentiating molecule. They are T4 cells. So T4 cells are having very important receptors are called major histocompatibility type 2. So the dangerous thing is GP120 goes and eradicate all the MHC. They damage the MHC. Now T helper cells without MHC2 cannot do anything. Just like a gun without a bullet. Understand? So the T helper cells cannot recognize foreign bodies. So many foreign bodies will come, but T helper cells cannot recognize. Humoral immunity will be failed, means many bacterial infections comes, and cell-mediating immunity is also failed. Many viral infections are also, T lymphocytes are also failed. So many viral infections also happen. So the body, a particular body where these changes occurred, the body welcomes all the bacterial, viral, fungal, protozoan infections. So one by one, all diseases comes and the person's immunity slowly, gradually decrease. Then a person acquired diseases. Acquired means what? Adopt one after one, one after one. Diseases will come and will not go. If a person get one disease, he will have one symptom. The person is getting all types of diseases, he will get a group of symptoms. The group of symptoms is called syndrome. So listen carefully. The victim is acquiring diseases. In the victim, diseases, nothing but immunity of the person is slowly, gradually decreasing, and group of symptoms are occurring, nothing but syndrome. That is the reason this disease is called acquired immunodeficiency syndrome. Acquired means what? Adopting all diseases. Immuno means what? Immune system. Deficiency means what? Decreasing. Then syndrome means what? Syndrome means not one disease. All diseases means many symptoms, group of symptoms. So AIDS means acquired immunodeficiency syndrome. Once a person detected he is a positive by symptoms, what kind of symptoms? Continued fever, coughing, weight loss, dramatically weight loss, and continuously dehydration of the body and motions. So severeness and headache, stomachache, so many physiological changes takes place in that body. Then that person is very near to the death. It is a fatal. This disease can be spread from that person to other person through injections. If there is no proper syringes were used, use and through syringes should be used. One syringe should not be shared from person to another person. Number two, why it is called sexually transmitted disease. This virus can also sustain in the fluid of a semen. In our body fluids also this is present, exists. So without condom or without any barrier methods, intercourse, sex happen, then from one partner to another partner, this virus can easily transmit. So it is called sexually transmitted disease. Not generally by intercourse this disease is coming. This disease can transmit through unprotected sexes, not by direct or regular protected sexes. So many measures will be there to avoid this kind of disease.
When HIV first enters the human bloodstream, the virus circulates throughout the body. The protective surface of HIV is studded with particular proteins called GP120, and inside is a capsid that contains the viral RNA and viral enzymes. Initially, the GP120 protein on the virus is only able to bind to a cell surface protein on the macrophage called the CD4 receptor. A second receptor protein, CCR5, must also be present on the macrophage for the virus to enter the cell by endocytosis. Viral RNA and enzymes are released into the cell's cytoplasm. Reverse transcriptase uses the viral RNA to synthesize first a strand of viral DNA and then the complementary DNA strand. The viral DNA then moves to the nucleus of the cell where it integrates into the host cell's DNA. Transcription of the DNA within the nucleus now results in the production of viral RNA. This viral RNA can serve as the genome for new viruses and can be used as messenger RNA to produce viral proteins during translation by the ribosomes. Complete viruses are assembled and released from the cell via exocytosis, which causes little harm to the macrophage. HIV cycles through macrophages over a period of years and continues to multiply while doing little apparent damage to the body. Eventually, the gene that codes for the GP120 protein is altered by mutation. The altered GP120 protein changes its co-receptor allegiance and now binds to a different co-receptor, CXCR4, which is found on the surface of CD4 plus T cells. The same processes occur within the T cells, resulting in new virus particles. However, as these new viruses leave the T cell, they rupture the plasma membrane, which kills the cell. As these newly released viruses invade and destroy other T cells, the body's immune response is weakened, and this leads directly to the onset of AIDS.
So this is how the disease is developing in our body. Now let us see how it can be diagnosed and how it can be treated. If you see a diagnosis of this disease, generally antigen testing will be there. Next, ELISA will be there. In biotechnology principles and applications, we see that enzyme linked immunosorbent assay. ELISA means what? Enzyme linked immunosorbent assay. ELISA. ELISA or ELISA. Assay means what? Test. And Southern blotting technique. Blotting technique is also helps. Western blotting technique to identify the viral load. So PCR, polymerase chain reaction, how much virus is present in. If virus is present or not, all these are okay. But how much virus is present, to check the viral load, the PCR will be used. At the time of PCR application, I said, I hope you remembered. And once diagnosis happened, what is the treatment? Generally, treatment is not there. Vaccines are under development, so that third generation vaccines are going over here. Third generation vaccines are going over here. Some in early stage the treatment will be there by azidothymidine. Azidothymidine, simply called AZT. If AZT injections will be given to the pregnant woman who suspected or who diagnosed as an AIDS patient or HIV person, then to avoid the infection of the pregnant, early stages if we give to the AZT, then through the placenta this transmission can be prevented. And also adults, individual adults, the very early stage if we detect the HIV infection, AZT will be the good medicine. So this is what the precautions. But awareness is so important. One should have very good awareness about the sex education, do's and don'ts, and reproductive parts usage as well as the maintenance, the changes that regularly and over the ages, what kind of changes occurs in our reproductive organs. One should have a very good idea so that we can easily maintain healthy and very important personal life also. And mainly in the adolescent, they do not have a proper idea about the reproductive organs, and they're not ready to even listen for some awareness camps and programs about the AIDS and all in the rural areas. So due to the lack of awareness, only these kind of things are happening. In NCERT, in our textbook, NCERT mentioned so many in our NCERT, in our textbook, NCERT mentioned so many things as a portfolio, do's and don'ts about the AIDS. And non-government organizations are also conducting so many camps to aware the people in the rural and tribal areas to about the sex education as well as these kind of STDs. So go through the lines of textbook and assign it. All the best.